day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. We have been working on an RNA-seq data set where we have been analyzing the data, right. The goal is to identify the genes that show difference in expression across two different conditions. Okay. So, just to remind you about the data very briefly. So, this is a data set with 16 samples and we have about 6,800 genes. Okay. And these 16 samples, these are organized in 4 groups and each group contains 4 technical replicates. Okay. So, if you remember, the groups are group 1, group 2, group 3 and group 4, and in the last class we have been looking at the differential expression analysis using DSEC2. Okay. So, this is the tool that we used, and if you remember, we followed all the steps starting from the count data. What we are going to do today is we are going to look at the same differential gene expression analysis, but with the spiking data in there. Okay. So, these are synthetic RNA molecules called ERCC spikins or ERCC spikin mix that will be used, right, for normalization. So, the data set that we have been working with, it also contains these ERCC spikins. Just to remind you again about the spikins that we talked about in the theory classes, is that these spikins, these are synthetic RNA molecules, they contain RNA molecules of different lengths, right, and they are added in different quantity or different number in each sample. Okay. Just to clarify that each sample will get exactly same amount of these RNA molecules. Okay. So, there is no technical variation here, right. So, there is no biological variation here. So, what you will get is purely technical variation, right. So, what you do is we add this exactly same amount of spiking to each sample, and at the end we get the read data, and if we see variation in read data, that will be technical variation, that will be due to technical variation, and we can then estimate the technical variation that arises because of the differences in sample preparation, etc. Okay. So, this will help us in differentiate between technical variation and biological variation better than the other methods where we have to assume some sort of distribution perhaps, or we assume that most of the genes are not showing any change in expression, etc. If you can also extend this using, when you are using these spikins, you can also estimate the absolute RNA molecule number in the sample, right. So, this is possible because you know the number of molecules of each type of synthetic RNA that you are adding, right. So, based on that you can calculate the RNA molecule of the sample that might be present. Okay. So, this is something again will not go into, but this is also more challenging because of the differences in the RNA sequence, right. So, there is also sequence specific bias, there are the other differences in GC content, etc. So, we will not go into that part, the absolute quantification. What we will work with is the relative quantification, right. So, relative abundance between two samples or two different conditions. Okay. So, this is the agenda for today's class. We will be working with DGE analysis where the data contains these ERCC spikins. Okay. So, again the steps, we have completed the first few steps that are shown in green, right. So, we have been actually working on the differential gene expression analysis. We have done the first differential gene expression analysis using DSEC2. Today, we are going to use the ERCC spiking data to do the same analysis and see if we get similar results or not. Okay. This is the agenda. So, the tools that we will be using are these tools. So, one is RUV-seq. Okay, and here is the link to the package. Okay, we will see in a moment where this package is and how we can install it and how to use this package. And once we have normalized using RUV-seq, we can then use DSEC2 to actually do the differential expression analysis, right. So, the second part we have already done, the first part would be the novel part. Okay. So, let us now move on to the terminal, okay, where we can look at the first look at the data, right, and let us find out, right, where this spiking data is. Okay. So, here we are in the terminal in the folder where we have the data and the codes, right. So, let us first look at the data file. Okay. This is the data file, if you remember correctly. If you see, right, there will be 16 samples from S1 to S16, and on the left side you have the gene names. Okay. So, the first column is the gene names, right. So, you have about 6,800 genes here, and then you have the samples names on top. So, this is the header line, right. So, when you load the data, we actually mentioned this is the header line, right. So, we have been doing that in R. So, what I will do is in this file now we can see where these spike-ins are. Okay. So, I will search for this ERCC, and you can see this, right. There are some rows, right, which are named ERCC 2, ERCC 3 and so on. You can see these numbers, right. So, these are the synthetic RNA molecules, these are not the gene names. Okay. So, all the gene names you can see they look different. These are the synthetic RNA or spike-in molecules that have been added to these samples. Okay. So, these RNA molecules, these are different RNA molecules, right, and they are also added in different quantity, right, to make the mix. Okay. Why different quantity? Because to estimate, right, how this variation in number, actual number and length can affect the technical variation or the read data count. Okay. So, that is why we have this combination of these different RNA molecules, not just one molecule. Okay. And then you can see these numbers, right, the abundance numbers, the reads that map to them is about 61,000, 5,200, right, so on, and for some spike-ins you can see the numbers are very low. Okay. So, these are present in very small numbers in the sample. Okay. So, you can see this huge variation, and then you can also probably notice that we have a lot of these synthetic molecules, and you can see these ends here, right. So, you have this ERCC 171. Okay. So, out of these 6,800 genes, actually not all of them are genes. So, we have about 100 or more, 150 or so, right. You can probably count how many are there are actually these ERCC spike-ins, okay, and rest of them are genes. All right. So, we can use this ERCC data now to actually normalize the data and can counter or minimize the technical variation, right. So, you get the idea. Hopefully, we have added these spike-in mix, right. So, each mix contains these multiple RNA molecules present in different numbers, okay, and we add this mix exactly same amount across all the samples. Okay. So, there is no variation due to environmental conditions, etc. When we are preparing the library, we have isolated the RNA, we add this molecule to these libraries, to these samples, okay, and then we get this variation in the data, which means this is purely technical variation that we can now look into the pattern, etc., and try to correct for this technical variation as much as possible. Okay. So, this is what we are going to do. We will import this data in R, and we will try to normalize the data based on these ERCC spike-ins. Okay. So, one of the things you probably now realize is that when you are doing the mapping in RNA-seq, okay, when you are building the index, you would also have to add this ERCC data, right, in the index. Okay. So, you have to have the ERCC FASTA files, those are available, and they will be combined with your transcriptome or genome data, reference genome data or reference transcriptome data, and the program will build index for that, right. Otherwise, you will not get this mapping against these molecules. Okay. So, this is something we need to remember. Okay. So, we will use this package DSEC2. We have I have shown you this in the last class. So, here is the bioconductor package, right, where you can go to this page and you can download, and we have also talked about, like, we can look at the manual here, right, reference manual, and we can find out the commands that we can use. The package that we will use today is called the RUVSEQ or RUVSEQ. So, as you can see, the name of this package is remove unwanted variation from RNA-seq data. Okay. So, this is what we want to do, right. So, we want to remove this unwanted technical variation that might be present in our sample, and the citation is given. This is the paper where this method has been proposed, or the package has been published. To install, this is a bioconductor package. To install, we will have to use this BioC manager, right. We have utilized this last time, if hopefully you remember. So, here it is, right, BioC manager install RUVSEQ. This will take a bit of time, okay. And in the meanwhile, we can Okay, so this is actually already there, probably I do not want to update any packages at this moment. So, it is telling me that this package is already installed. Okay. So, if it is not, then it will install step by step. Okay, so what we'll do is, now we'll have a look, right, what this package can do, right, and we have the manual around here. Okay, so I'll click on this link, and you can have all the commands, right. These are the commands that are available for this package. Okay, and we'll use one of these for our data. Okay, and again we have all the descriptions, all the attributes that a command can take, and also sometimes some examples, right. So, the usage, attributes and examples will be given. The method that we will use so there are three methods you can see at the top. If you go, you have this roofg method, you have roofr, and you have a roofs. Okay, so, what are these? Let us have a look, right. We can click on this link. So, this is roofg, right. So, the full form you can see here, remove unwanted variation using control genes. Okay. So, if you have a set of control genes that you know, okay, these are not differentially expressed between samples, you can use that set for normalization. Okay. So, this is the roofg method. Then of course, you have this roofr, right. So, again this is remove unwanted variation using residuals. Okay. Here you do not have these control genes, so you do not have to specify that, and the last one is roofs. Okay. This is remove unwanted variation using replicate negative control samples. Okay. So, this is again a different method, again different requirement, but what you probably have realized now is that in our case we can use this roofg, right, because this removes this variation using control genes, and we can consider these spike genes as control genes, right. So, these are control genes, mean they do not show any difference in expression across samples, right. So, there is no biological variation, and so ERCC spike genes, they fit that criteria, right. So, we are not seeing any, we should not see any variation in this ERCC data across samples, right, because they have been added later on after we have isolated library from the samples, and they have been added in equal amount. Okay. So, we can use this roofg, and we can say, okay, we can use this ERCC as the control gene. So, we just have to specify these ERCC genes as the control genes to this method. Okay. So, this is what we are going to do. Then we have the usage that is mentioned, right. So, you have roofg, you have these attributes that are given. This X is usually a gene by sample matrix, or it could be a seq expression set. Again, it can take a specific data format, right, and we have seen how we can create this seq expression set in the last few labs, I think, right. So, when we are doing this hands on, we have seen we have created this seq expression set data format using this new seq expression set command. Okay. So, then we have these other attributes that we also sometimes need to specify, right. For example, whether you have rounded normalized measures, right, or you have epsilon. So, if you want to add this number, right, before you log transform, because sometimes if your count is 0, right, you cannot take log 0. So, you can specify what would be the number that you want to use, right, and so on, right. So, there are so many things, right, and then you have the k, that is the most important part, right. This is the number of factors of unwanted variation to be estimated from the data. Okay. So, we can probably end up with just one or so, right. So, we can say 1, 2, etc. We'll see, like maybe we can just specify with 1, and we'll try out this method, and what I'll tell you is that you can try different case and see how that will change the results. Okay. So, then there are some detailed explanations, as you can see right here. It clearly mentions several types of controls can be used, right. So, you can use housekeeping genes if you know you are sure, right, these are not showing any difference in expression, or you can use spike genes. Okay. So, it specifically mentions ERCC spike genes, and we have these ERCC spike genes in our data. So, we can use those for this roofg method. Okay. And then of course, there are other, so here are references, and then the example set. Okay. So, we'll use these commands. Now, we can see this, right, how this process will go, and we can identify, or we can try out this method for our data, and once we have done that, then we'll use the DSEC2 to do the differential expression analysis. Okay. So, let us try the first part, and as you can see here in this example they also used k equals to 1. So, we will try k equals to 1, we can increase this later on, you can try it yourself, right, when you are doing this. Okay. So, again what I will do is I have actually collected these commands, right. So, we have this data, right. So, let's go to this command line. Okay. Here we are using this RUBSEC package, we will load the data and then we will complete the analysis. Okay. So, the first part will be the new part, then we will do the differential expression analysis using DSEC2. So, that is something you are already familiar with, we have done this in the last lab. Okay. So, I will load the library first. Okay. So, library RUBSEC. So, it will load, it will give us some output or some like commands, right, and then we need to load the data, right. So, here is the data WH data which I am going to load. Okay. Okay. So, here it is, it is giving some command, right, some comments, right, it is saying it is masking some objects, etc. So, we do not have to worry about it, there is no error at least. So, what I am going to do is I am going to load the data. We have the header, so we have to give header equals to true. Okay, and the data is loaded correctly, you can see by this head command, right. So, we have the sample names, so which are the column names, and then you have the gene names which are the row names for our sample. Okay, so, the next step is to define what are these control genes or spikes. Okay. So, from this data, right, we have to mention, right, okay, these are the rows that represent the control genes, okay, and the corresponding counts. Okay. So, this is what we are going to do here in this command, as you can see this is what we will do. Okay. So, spikes row names WH data. So, we are actually getting the row names of the WH data and matching, okay, with this grep ERCC. Okay. So, grep ERCC means is that we are matching this word ERCC, right, for the gene name or row name in the row names here, right. So, this command is searching for this grep ERCC row names WH data, right, it is searching for this data in the in this WH data, and where there is match that will be stored as spikes. Okay. Those row names will be given as spikes. Okay. So, let's try this command, okay, and see what we get, and then you will realize how this actually works. Okay. So, this first part, this part here, it is looking searching for this ERCC in the row name, okay, and the second part is only taking those row names where this match is found. Okay. So, maybe I can try this out separately. You can see, right, if I write grep ERCC row names WH data, okay, it is giving me the row numbers where it is finding this match, okay, in the data, and the spikes we can now try, okay, it gives us the names of these spike ins. Okay. So, these spike ins that are present in the data, we have now identified those using this very simple command, using the grep command, right. So, this grep command we have talked about in the terminal, and this is what we use here. Okay. All right. So, once we have now defined the spikes, what we are going to do is we are going to set up something called as factors. Okay. This is simply defining the design of the experiment. Okay. So, what are these factors, right. So, this is simply saying, okay, so the order of the samples and their which condition they belong to. Okay. So, in our case the first 4 are group 1, the next 4 are group 2, the next 4 are group 3, and the last 4 are group 4, right. That is what we are defining here, right. As factor we are defining this variable x, and as factor, right, we are making this list from, and the order of this should match with the name of the samples. Okay. So, it is simply getting this order of the samples, right. So, which group they belong to, and the 4 levels, as you can see, it is defined the G1, G2, G3 and G4, right, 4 factors. Okay. So, this is what we have done now. Now, what we are going to do is we are going to create this expression set. Okay, this is something we have done before, and here we are going to do the same thing. Okay. So, this is the command, right. If you go back now, how to create this seq expression set, we have this count data part, we have the pheno data part, right. So, again go back to the last class, or maybe go back to the file, right, how to create this new seq expression set, right. You will see we need the count data and the pheno data, right. So, that is what we are doing, and we are assigning this variable set with this data. Okay. So, I will run this in here, okay. And so, this is simply coming from the whole data that we have loaded, right. This is the as matrix form, that will be the count data, and then pheno data is the in the data frame format, right. So, it is a x, right. So, the x is the factors that are given, right, the groups that we have defined just now. Then we have the row names. Okay. So, the row names will be the column names of this data, right. So, column names of this data are the sample names. So, these sample names would be the row names of this seq expression matrix. Okay. And let us see, we have done this now, and then if we look at set, it will tell you, right, this is seq expression set format. We have 6,800 genes. Of course, this is not genes, this includes ERCC. We have features, right, 16 samples, counts, normalized counts, element names. If we have not defined the normalized counts in here, so we do not have to worry about that. We have 16 samples, S1 to S16, variable labels are X. Okay. So, this is okay. This is we have defined now this set. Now, once we have defined this set, we can now run RoofG. As you can see, RoofG requires a new seq expression set data, right. Here you can see this in this example. It requires a new seq expression set data, which you have defined, the list of spikes and the number of factors. Okay. Of course, there are other attributes, but these are actually required. Even I mean, these are mandatory, right. You have to mention how many factors you are looking at, which are the spike genes, and what is the data that you are looking at. Okay. And these should be enough to run your RoofG method. Okay. So, that is what we are going to do, right. We are going to run this RoofG method now. Okay. And let us go to the terminal. Okay. Sorry, set 1. Okay. So, it has run this RoofG method now. Okay. And we have got this set 1. Okay. So, what we want to do is we want to see, right, whether set 1 is slightly different from set at all, right. Is there any difference in the data that have been obtained, right, after this RoofG method. Okay. So, this is something we will do first using this plot RLE method. Okay. So, plot RLE will tell us, but before we do that, I will define this color set. Okay. So, this is simply saying what would be the colors of these boxes in the RLE plot. Okay. So, if maybe I can just run this RLE plot, and the set 1, this is the data that we are running. Then we have, we do not want outline, y limit, right, that is the limit of the y axis that is given, the range. Okay. We can simply run this, and then we will see, right. Okay. You can see this will be in gray, right, or this the whole box plot is shown in gray. You have these samples here. So, it would be nice if we can define some colors for these groups, right. So, if we can group them by colors. Okay. So, to do that, we will define this colors variable. Okay. And we are using 4 colors. You can choose anything you like. So, this is red, blue, green and chocolate. Okay. So, red, this means you have 4 reds, like, so these are group 1. So, if you see the order, we have 4 group 1 samples. So, this would be 4 reds, then you have 4 group 2, then 4 blue, then you have 4 group 3, 4 green, right, and then 4 group 4, 4 chocolate color. So, you can define any color you like. I am just defining them. Okay. And then we run this plot RLE again. Okay. But this time color with our colors that we have just defined. Okay. And you can see this now, these samples are colored. Okay. So, it's the red one is for group 1, the blue is for group 2, right, and green is for group 3, and the fourth one is for group 4. Okay. Now the question is there any difference with the original data? So, let us check that. Okay. Let us run this with instead of set 1, let us run this with set, right. Set is the original data that we created. This is a new sec expression set that we created. So, let us run this with that new sec data, right, and see if we can see some difference, if we can observe some differences, right. So, here is the original data, the raw count data, okay, and this is the RLE plot. That is why the y axis is transformed, and then if we plot the set 1, again you can see that the difference between the raw data and the normalized data. Okay. So, now we have done this, we have done the normalization. Okay. So, it is complete, and in the next step we can do the dsec2. Okay. So, that part you are familiar with, right, we have done this before. So, we have to load the library, right, dsec2 library, then we have to create this dsec data set from matrix. This is again a command that we have to use because we need this dsec data set for running dsec2. The count data now would come from this set 1 matrix, right. So, not the set matrix, but the set 1 matrix. This is the normalized count data, right, after the roof G normalization. Okay. So, this will come, and then we have the column data that will also come from set 1 and the design part. Okay. This is very important here. Okay. So, this design part actually if you look at this P data set 1. So, we can write set 1. Okay. So, when you see the normalized data here, okay, so set 1, you see there are two variable labels, right, x and W1. Okay. So, W1 is the factor, right, this factor that has been estimated by roof G. Okay. So, you remember, right, how many factors you want to estimate from the control genes, right. So, this is the factor. If you actually estimate 2 factors, you will get W1, W2, 3 factors, so on, right. So, since you have estimated this factor, this should be also part of your design, okay, and x should be part of your design because x determines the groupings, right, which sample belongs to which group. So, we will have these 2 variable variables, right, and this will be part of the design in dsec2. Okay. So, let us clear this window now, and let us first load this library dsec2. Sorry, yeah. So, we need to mind the capital letters and the small letters. Also, I will close this. Okay. All right. So, now we want to create this dsec data set, right, from matrix after this roof G normalization. Okay. The difference is the counts will come from the normalized data and the design will be different. Okay. So, the design will consider both these factors, right. So, the x is the group, right, that will be define the groups, and W1 that has been estimated by the roof G method. Okay. So, this is the command for. Okay. So, we have now created this data set, right, dsec data set, and as you can see as before, right, we have this dsec data set class, dimensions are given, 16 samples, 6,800 genes, right. Then you have these row names that are given and the column names, these are the sample names, and then we have these 2 factors that are also given. Okay. So, now it is easy, right, once you have run this, once you have created this, right. So, we can now run dsec, okay, on this, it will again give you the steps, right. So, we have discussed these steps, estimate size factors, then dispersions, right, these alpha i's in the theory class if you remember. Then you have these gene wise dispersion estimates. Okay. So, those will be the I running for all genes. Then you have mean dispersion relationship, which is used for dispersion shrinkage, then that will give you the final dispersion estimate. Once you have this dispersion estimate, you can fit this model, this negative binomial distribution, and then you can do the test. Okay. So, the testing is the world test. Okay. So, here it is, right, again the size factor is now determined for these samples. Okay. So, the simple part now is to look at the results. Okay. And we create these results by this command, and we can simply say res. So, we get this base mean, log fold change, the statistic, p value and p adjusted. Okay. Now, this is as before, right. You can go ahead and do the same analysis. So, what I will do is I will simply plot this now, okay, and this MA plot will give you this log fold change versus mean counts, right, and we got something very similar in the last class if you go back. Okay. There would be some changes of course, but you can see this looks pretty similar. Only one thing that we can probably do to actually make it better is the ellipsis shrinkage, right. So, you remember this again from our last class. I am skipping some of these steps perhaps because we have done this before. Okay. So, we will load the library APGLM, we will look at this PRDDS and then we will do the ellipsis shrinkage and we will again plot, right, and we will see, right, what are the results that we get from here. Okay. So, loading this library APGLM, and once it is done we can, okay, so it is telling us, right, this design thing intercept W1 x G2 versus G1 x G4 versus G1. So, this is not bean anymore, right. So, that was in the earlier class, right. So, we have seen, so this will not be bean. So, we will use this command to generate this these results after ellipsis shrinkage. The coefficient would be x G4 versus G1, right, and the type would be PGLM. We can use other methods as well, and once this is done we can plot MA and see if that looks okay. Okay. So, this is done, we use the plot MA command and we get better results. Okay. So, one thing you probably have to do now is actually compare, right, how this goes going back to the earlier DSEC2 analysis and see how these results look. Okay. There would be some differences, they look very similar, but there would be some differences, especially when you export the data, and also if you can look at this data here, you will see there is probably some difference. Okay. So, I can plot this. So, if I remember correctly, right. So, for this gene, right, in the earlier analysis it was slightly different value for log fold change, and this has changed. This will change because we are using this correction or this normalization using this root G method. The rest of the things you can then now filter, right, and identify the list of genes. You can export them to a file, right, in a text file, tab separated file, and you can work with those files. Okay. So, this completes our differential expression analysis, and what we want to do is beyond this MA plot we want to look at some visualizations, right, for example, the volcano plot or the heat map, and we also want to look at the gene set enrichment analysis, right. So, the function enrichment analysis we have talked about. So, very briefly I am to show you how we can do that. There are different tools, but we will use one tool and we will complete that analysis, but that is for the next class. Thank you very much.